All right. Okay. We are back. We had seen the title. Previously, we had looked at a recap of what we had yesterday, the other two experiments. Then today we have the experiment to demonstrate that carbon dioxide is essential for photosynthesis. Now, like it has been read, we need to get two healthy potted plants, which are almost the same size. And then we place them in the dark for 24 hours. Can I know why we have to place them in dark for 24 hours? Someone to respond. Why do we have to place the plants in darkness for 24 hours? Yes, please. Fiona? Yes, Anastasia? Anastasia? Yes, Anastasia.
To discharge the leaf.
Very nice. That is very nice. The reason we keep in darkness is to discharge the leaf, okay? And discharging, I told you yesterday, it means removing starch. So when you keep it in 24 hours for that period of time, all the starch that is there is removed. So that by the time you start this experiment, all the leaves in the plant have no starch. You don't have to be biased by you going with the leaf which has starch, and yet you are testing if starch is there when it was there before. I think now you understand the reason why we have to discharge before the experiment is started. It is very important to first keep the plant in darkness for 24 hours. So when you're describing such experiment on photosynthesis, you have to be very keen on this, making sure you have discharged the plant. Okay, all right. Then now yes. We place the plants and cover them separately, like you see in the setup here. We cover them separately. However, we keep some crystals of potassium hydroxide in a petri dish. You can see. I want to. Mute Fiona. Okay. All right. I think now we can follow. Like we see there in the setup, we have put potassium hydroxide crystals in the petri dish. And then in the other, we see a barrel jar without potassium hydroxide. Now, the gist of this experiment is all about carbon dioxide. We're testing to see, the question is carbon dioxide essential for photosynthesis? That's why in our design, we emphasize looking at how can we have carbon dioxide in the setup and no carbon dioxide. And in this case, to remove carbon dioxide from the setup, like in the first setup, that is why we have potassium hydroxide crystals in the petri dish. This potassium hydroxide, so this potash, what it can do is to absorb carbon dioxide all around. And remember, we have said that we ensure that this barrel jar is airtight, yeah? Airtight by applying Vaseline. So all along here, if you can see where I'm moving my cursor, all along there we put what? We put Vaseline to make this setup air what? To make it airtight, so that no air from the outside gets into here. All right. So we only maintain the air which is enclosed within this barrel what? This barrel jar. This is the barrel jar. This barrel jar, you can see it. Okay. So in the next setup, we have not put potassium hydroxide, which means this is the setup with all now the conditions for photosynthesis. And in this one, we have eliminated the condition that we are testing for, and that condition is carbon dioxide. So at the end of it all, we want to see if really this plant which had carbon dioxide and only had carbon dioxide, which of them was able to photosynthesize. If we find out that this one will have to starch, the first one, then that means that if there's no carbon dioxide, such photosynthesis can take place. If we find there's nothing but is here, that means shall conclude carbon dioxide is an important component, requirement for the process of photosynthesis. So that is what we have in the setup. So we have arranged those two. Remember, one of them is the real experiment. The other one is the control experiment in that case. Now, after making such arrangements, we keep the plants in sunlight for photosynthesis to take place. And this can be done. We can leave them in the sun for 3 hours. Although in the write-up, we are saying 3 to 4 hours. It is always good to be specific on a particular period of time. Like you have to say for 3 hours. For you as a student, if you are describing the experiment, you have to be specific and tell us 4 hours or 3 hours. Don't say 3 to 4 hours. 3 to 4 hours is in the notes. But if to be practical enough, you have to tell us the period which you have used for you if you're describing the experiment. That's a common mistake. When people are writing, again, like in the next exam, they say, leave it for 3 to 4 hours. That's the sort of thing. What did you choose out of the two? This is just notes, so we can accept that. But for writing purposes of experiment, if you're doing it, you have to tell us what period have you used. That point, I think, has been noted. After which, you pluck a leaf from each of the plants. Remember, there are 2 setups, one with the carbon dioxide allowed in, one without carbon dioxide. Then, but then you boil the leaves in water and subsequently in alcohol using a water bath to remove chlorophyll. This is the procedure we took yesterday when we want to test a leaf for starch. You get it, boil it in water, then alcohol, then you dip it in water. Okay, and the reason why you boil in alcohol is to remove chlorophyll. Then later we add iodine to that leaf in question, and then we take our observation. Now, the result for this experiment show us that only one leaf turns blue-black, and this shows the presence of starch. When you see blue-black, it means starch is present, and this happens because potassium hydroxide absorbs the carbon dioxide present in one of the biologia. The second biologia, we didn't put potassium hydroxide. That means carbon dioxide was not tampered with. But with this first one, there was tampering with the carbon dioxide present because it was absorbed by potassium hydroxide. So as a result, the leaves do not get carbon dioxide for photosynthesis, and that's the process of photosynthesis is inhibited and the starch is not synthesized. Why? Because the potassium hydroxide, like in the first setup, I'm talking about this first setup, the carbon dioxide was all removed by this potassium hydroxide. So when this leaf was left with an option, maybe it was having other requirements for photosynthesis, there was light to add its own water. It had been watered very well, but now the component of carbon dioxide was missing. So don't expect a reaction to take place, and photosynthesis cannot take place in that case. But for the second setup, where the carbon dioxide was there, light was there, although in your setup it is not showing it, but light is there. We don't normally draw the sun and arrows to show there's light. There, the leaf got from that second setup, we realized that for it has starch. Tested positive for what? Tested positive for starch. Okay, so we later prove or conclude that really carbon dioxide is needed for photosynthesis to take place, because when we eliminated it in the first case, the leaf did not carry out photosynthesis, and where we left it, the leaf carried out photosynthesis. Altogether, class?
Yes.
Anything? Have you been following?
Yes.
Yes.
All right. All right.
Yes.
Okay. Now that you have been following,
Yes.
I normally bring in
Question.
with some questions to show that people have been
Yes.
following. And the first question will be how do we, okay, question, let me repeat the question. So it should be, what is the role of potassium hydroxide in this experiment?
Pardon.
Question, I've written the question in the chat.
Pardon.
The question is typed in the chat. What is the role of, are you able to see the chat? What is the role of potassium hydroxide in this experiment? Hello? Hello.
To absorb carbon dioxide.
Very nice. The role of potassium hydroxide. Yes. The role of, very nice. The role of potassium hydroxide is to absorb carbon dioxide from within the barrel, the barrel jar. Okay, that is nice. Second question. Second question. How did we? How? Okay, so question is, why was the plant? Why were the plants? Why were the plants discharged before the experiment? The question is written in the chat. Why were the plants discharged before the experiment? Yes, please. Trevor? Trevor, give us the answer.
For me, I think.
Yeah.
Side.
Hmm?
Hello?
Yes.
For me, I think.
Yeah.
Reduce on the rate of carbon dioxide.
Sure. Can we have another try from Peter? Peter Clever?
Teacher, for me, I think that since the experiment is about photosynthesis, the leaf had to not be having any starch such that when we bring it out at photosynthesis, it has somewhere to do it from.
Okay, very, very good. So the plants were actually kept in darkness for 24 hours to discharge them, so that by the time of the experiment, all the starch has been removed, okay, to avoid bias. You get, like, if you don't discharge and then you bring them in the barrel jar. Yeah. Now this one where you put potassium hydroxide, already the starch will be there in the experiment. That's why we first remove starch in all of them, okay? And we bring them when they are free of starch by keeping them in darkness for a period of 24 hours, okay? Then maybe my last question on this. Why do we need a control experiment? Why do we need a control experiment in this case? And what is that control experiment? Why do we need a control experiment? And what is that control experiment? Yes, please. Yes, please. Can I choose someone? Rosemary Namakula. Rosemary Namakula. Sechide. Segawa, today I've come early. Yesterday you came when you are almost closing. Agaba. Joy. Anyone ready to give me the answer? Now, yesterday I was telling you that whenever you are designing an experiment, in order to make valid conclusion, you have to design a control experiment. Like in this case, if we are having a plan where we are removing carbon dioxide, then we have to arrange another one where we have all the conditions available. Yeah, we have provided all the conditions. Okay, the carbon dioxide must be present. So we see when there is no carbon dioxide and when there is carbon dioxide. So the control experiment helps us to make a valid conclusion of the results of the experiment. You cannot think your results are valid if you have not designed a control experiment. Hannah Peace, you have just joined. Before you joined, we are looking at an experiment to show that to demonstrate that carbon dioxide is essential for photosynthesis. And before you joined, you had read through this script as shared on your screen there. We had seen the importance of each of the component, especially potassium hydroxide. We had seen the reason why we have these two plants dispatched before, and why we have to have two setups for that experiment. So, and then we say the control experiment is a setup, okay, that is meant to ensure that we make a valid conclusion of the results we have obtained. How we arrange an experiment in such a way that if, for example, in this case, carbon dioxide had been eliminated by putting potassium hydroxide, in the control experiment, we find a way of ensuring there is carbon dioxide not tampered with, yeah? So that we see what happens to the plant where there was carbon dioxide and the plant where there is no carbon dioxide, because our experiment, we want to see if carbon dioxide is essential for photosynthesis. So we must have a plant where there is no carbon dioxide. That is our main issue here, okay? Then if our main issue is to test for presence, if the essentiality of carbon dioxide, is carbon dioxide really essential? Yes. So what we do, we remove it and we see if this plant will carry photosynthesis. Then in the other case, we ensure carbon dioxide is there. Why? We have not put, that's why you have beroja without potassium hydroxide. So that means carbon dioxide is going to be present in that, is going to be present in this experiment, right? And here, there is not going to be, this particular one, there's not going to be carbon dioxide because there's potassium what? Hydroxide. You can use anything else which you know can absorb carbon dioxide. Even sodium hydroxide, yeah, can what? Can absorb. If you provide some experiment, you find I've used the maybe soda lime, yeah, it can do the same. The reason why we are putting these components, most potassium hydroxide, sodium hydroxide, these are good at absorbing carbon dioxide, but this one is most effective. Okay, potassium hydroxide is the most effective, is the more efficient in absorbing carbon dioxide than any other of those. We can use, even lime water can do it. But now here we choose one which is the best. Okay, so potassium hydroxide is the best in it can absorb more faster compared to the other. Okay, sodium hydroxide can do the same. even calcium hydroxide, or what we're calling lime water. If you put there lime water, it can do the same role. So you'll find that some books will have different setups using different components, but the reason is one, to absorb carbon dioxide, whatever you use, okay, as long as you can explain it. I hope I've tried to help up my friend who was saying that was Hannah, who had just joined, and I hope you have been able to follow up by the end. By the time you found us, maybe we had even looked at some questions. You found out at question time. However, we are meant to continue to the next piece of work for this. And this is the second experiment. Can someone read for me the title for the second experiment you are looking at for the day? Yes, please. Yes, Hannah, please.
Experiment to show that sunlight is essential.
Experiment to show that sunlight is essential for photosynthesis.
Very nice. Can you go through the materials required?
Potted plants. Aluminium foil.
Let's have one person read. It was okay. Okay, Sekide, go on with the materials. Sekide, Mark, go on with the materials. Yes, Mark. Hello.